Good morning. In this video, we are going to discuss the cell membrane proteins and their different structures and functions. But before doing this, let's remember together the fluid mosaic model of the cell membrane. As you know, the cell membrane is a double phospholipid membrane, meaning that it's made up of two layers of phospholipids. And we have seen before that in this first layer of phospholipids, the phosphate heads are usually or always actually directed outwards. And in the other layer, the phosphate heads are always directed inwards towards the cytosol of the cell. So here they are directed outwards towards the extracellular fluid, the fluid around the cell, and the innermost layer has the phosphate heads directed towards the cytosol. What's common between the extracellular fluid and the cytosol is the presence of water. So the extracellular fluid is water plus dissolved substances, and the cytosol is also water plus dissolved substances. And that's because the phosphate heads, as you know, are charged. So they are polar, and polar likes polar. And since water is polar, so these phosphate heads are polar and are always attracted to water, as we said before. The cell membrane is, or the model that has been proposed by the scientist Singer, described a model of the cell membrane called the fluid mosaic model. Why is it called the fluid mosaic model? Because actually, as we said before, if the cell membrane was only made up of the phospholipid bilayer, we would find that the cell membrane would be rather fluid with the two layers, this layer and that other one, like sliding over each other. So in order to minimize that sliding, we should anchor molecules every now and then along the cell membrane, as you see here in the diagram. So here, for example, you can see a glycoprotein molecule. Here you can see a protein, which we call a carrier protein, and we will see why. And then we have another channel protein. And then on the inside of the membrane, we have another protein. That protein is considered a peripheral protein. And peripheral here is in contrast with the term peripheral here is in contrast with the term integral. When we say an integral protein, we mean that the protein molecule takes the whole thickness of the molecule. It takes the whole thickness of the molecule. Whereas when we say a peripheral protein, we mean that this protein does not extend along the whole thickness of the cell membrane. And the cell membrane, because of the presence of these different types of proteins, in addition to cholesterol, as we said before, these molecules diminish the fluidity of the cell membrane, and meanwhile, they control, especially the protein ones, control the passage of substances across the cell membrane, as we said before. So the cell membrane is considered, or this model is considered mosaic, meaning that you have a mixture of different types of molecules. It's not a homogeneous structure, but it is rather a heterogeneous structure with different types of molecules inside the cell membrane. And the cell membrane is considered selectively permeable. Permeable means allows the passage of. Selectively permeable means that it controls and selects what molecules or substances to pass and what substances will be prevented. This fluid mosaic model is actually a model for eukaryotic plasma membranes in particular. So eukaryotic plasma membranes are represented by the fluid mosaic model, which indicates that we have a phospholipid bilayer with proteins that are dispersed along the surface of the cell membrane. Because the phosphate heads are hydrophilic and the lipid tails are hydrophobic, we say that the phospholipid molecule is amphipathic, meaning that it has both characteristics. Part of it is hydrophilic and part is hydrophobic. What are the cell membrane proteins? As we said before, the first group of cell membrane proteins are the integral proteins. And when we say that they are integral, this means that they completely span the thickness of the cell membrane. And therefore, these integral proteins, though being proteins, and being mainly mainly hydrophilic, they will have to deal with or to manage passing through the hydrophobic interior of the cell membrane. What is hydrophobic inside the cell membrane? Yes, the lipid tails, because they are made of lipids. So what happens is that those integral proteins should have, for example, if we talk about the protein channels, like the sodium channel that allows the passage of sodium ions, because sodium ions are charged, so the inside of the channel should be made of amino acid residues that carry charges or that are polar, and it has to be hydrophilic. But the outside of this sodium channel, which is a protein channel, will be in contact with the lipid tails. So those outside portions of the channel that are in direct contact with the lipid tails should be hydrophobic. And this is how an ion channel or a channel in general helps the passage of polar substances or even charged substances by having hydrophilic internal residues of amino acids to allow the passage of the ion or the charged molecule. And on the other hand, from the outside, mainly it will have hydrophobic areas to match with the hydrophobic or to interact actually with the hydrophobic tails. But we also have peripheral proteins, as we said before, and those proteins do not fit into the whole thickness of the membrane. They are just like this purple protein, this one. They're just attached loosely to the surface of the membrane, and they consist mainly of glycoproteins. And as we said before, glycoproteins are essentially protein molecules. So the prefix glyco is what we add to the protein. So it is essentially a protein molecule to which a carbohydrate moiety has been attached through a covalent bond. Usually, these peripheral proteins that are glycoproteins serve as signaling molecules. Signaling molecules in what sense? Does this mean that they come from other cells to signal the cell? No. Here, the glycoproteins serve as signaling molecules, meaning that they help distinguish one cell type from another cell type. So blood cells, for example, will have peripheral glycoproteins of a particular structure. Skin cells will have peripheral glycoproteins of other structures. And the best example is blood grouping. Blood grouping is determined by certain signaling molecules or antigens on the top of the cell membrane that determine, for example, or differentiate between blood group A, blood group B, and so on. So glycoproteins on the surface of the red blood corpuscles are responsible for the ABO grouping. We group the blood types of human beings into blood group A, blood group B, blood group AB, and blood group O. So if the red blood cell has the glycoprotein antigen A, then the blood group of that individual will be A. If it carries B, glycoprotein, B is just a symbol that is given to the glycoprotein, then the blood group will be B. If it has both, then it will be blood group AB, which is a bit less frequent. And if it has none of the antigens, then it will be blood group O. And we will be seeing later on in genetics how the presence of such antigens determines which blood groups can be transferred to which blood groups and which ones cannot be transferred. For example, it is well known that blood group O is a universal donor, meaning that it can be transfused to any other blood type. But blood group AB is a universal recipient, meaning that it can receive blood from any other blood type, but donates only to AB. Some of these cell membrane proteins, we usually call them cell membrane proteins, whether they are integral proteins or peripheral proteins, whether they are glycoproteins or other types of proteins, they help attachment to the cytoskeleton of the cell. For example, here we can erase all the editing. You can find, for example, a particular protein here to which a microtubule or part of the cytoskeleton of the cell is attached. While there are other proteins that drift laterally along the cell membrane. So you will find some proteins that are kept in place by those cytoskeletal fibers, okay? Whereas other proteins that are not attached to the cytoskeleton might be displaced to the right and to the left laterally. We call this drift because the cell membrane is essentially fluid in nature. The fluidity of the cell membrane, we've just discussed this, and we've just said that phospholipids move along the plane of the cell membrane rapidly, and that cholesterol molecules are embedded mostly in the interior of the bilayer to stabilize the cell membrane. And the cell membrane consists of phospholipids, but there are embedded in the phospholipid bilayer proteins. Imagine that those proteins that are embedded in the phospholipid bilayer represent 60% of the mass of the cell membrane, and they are responsible for this mosaic appearance of the cell membrane, whereas the lipids represent only 40%. As we said before, number 4, the external surface of the plasma membrane has glycolipids that are we talked only about glycoproteins before, but now we are talking about something additional on the surface of the cell, the outer surface of the cell membrane, which is the glycolipid. A glycolipid is essentially a lipid molecule to which is attached a carbohydrate moiety. So it reminds us of the glycoproteins, like the ones that are present on the cell membrane of the red blood corpuscles, and that helps in ABO blood grouping as well as RH grouping. RH is another type of antigens that is also present on the surface of the red blood corpuscles. 80% or 85% of the population of human beings are RH positive, meaning that they have this antigen, and 15% do not have it, and therefore they are RH negative, and it also should be present. It should also be taken into consideration when transfusing blood and during pregnancy, because it's very dangerous if the mother is RH negative and the fetus is RH positive. Let's go back to the glycolipids. The glycolipids are again lipid molecules to which a carbohydrate moiety is attached, and again attached by a covalent bond. And we will see that the glycolipids also help in cell-to-cell recognition. So we have two types of molecules on the external surface of the cell membrane that help cell-to-cell signaling and cell-to-cell recognition, which are the glycolipids and the glycoproteins. And notice that the carbohydrate moiety is common between the two. Let's now see an overview or a wrap-up of the functions of the cell membrane proteins. The first function of the cell membrane protein is the transport functions, because we have just seen that we have membrane channels and, if you remember, and membrane carriers. So we will be discussing the function of channels and the function of the carrier proteins. The channels are essentially important for ions. So we have, channels are plasma membrane proteins that are important, as we said before, for the transport of ions. So, for example, we would have a sodium channel, we would have a potassium channel, we would have calcium ion channels, and so on. But we also have, in addition to the channels, we have carriers. And carriers are important for the transfer of large molecules, especially if these large molecules are polar molecules, like, for example, molecules of amino acids and molecules of glucose. And the major characteristic of those carriers is that they can change shape. So they first accommodate the molecule like this, glucose, for example, and then they, using some kind of energy, the two parts or halves of the carrier will change their direction in order to allow the glucose molecule to enter. This is, of course, an oversimplification of the process. We will be studying this in detail. But in addition to the ion channels and in addition to the carriers, we also have pumps. There are some of these protein molecules that help active pumping. So when I talk about a pump, I mean a protein molecule embedded in the cell membrane that helps active transport of a molecule from a place where it has lower concentration to a place where it has higher concentration using energy derived from ATP. And then finally, we have the electron transport chains. And it is worth mentioning that the electron transport chains are mainly present on the inner membrane of the mitochondria, as well as in chloroplasts on the thylakoid membranes. They are again protein molecules that help transfer of electrons from one electron transport carrier to another, but they meanwhile act as pumpers of protons against concentration gradient. But for the moment being, we have to remember only that they are proteins. So the electron transport chain is a chain of protein molecules embedded in the inner mitochondrial membrane or in the thylakoid membrane in the chloroplast that help transfer of electrons. But the cell membrane proteins can also have specific enzymatic function, and we will understand later on how the enzyme adenylate cyclase enzyme that is bound to the cell membrane helps converting ATP, which is not a cyclic molecule, into cyclic AMP. Cyclic AMP is important. It has a totally different function. ATP is there to be hydrolyzed into ADP and phosphate and energy. So it is a source of energy, but cyclic AMP acts as a secondary messenger inside the cell. And we said this before, and we will explain it explicitly in the unit on cell division, or even we might explain it at the end of this unit. If you have a molecule that cannot enter the cell membrane because it is, for example, hydrophilic, charged, and so on, and that signaling molecule attaches here to a receptor, this will stimulate the formation of cyclic AMP, and cyclic AMP will do all the chemical changes that this original messenger or signaling molecule wants to do. And we will be studying this in detail. The third important function of the cell membrane proteins is signal transduction. We have just talked about receptors. These receptors may receive signaling molecules, and those signaling molecules may be, for example, hormones. They may be neurotransmitters in the brain that transmit information from one nerve cell to another. They might be cholesterol. So these receptors, as I said before, help converting this binding that occurred with the signaling molecule into an internal change inside the cell, and this is what we call signal transduction. So in number 2, while explaining adenylate cyclase function and cyclic AMP function, we explained signal transduction without formulating it into words. So what we have explained in number 2, which is the ultimate formation of cyclic AMP resulting in chemical changes in the cell, is the explanation of signal transduction. And signal transduction results in changes inside the cell, which we call an intracellular response. But we also have seen that those cell membrane proteins can help attachment to the cytoskeleton. We saw before in the last video that they help attachment to the extracellular matrix. And when we drew the diagram of binding through the cytoskeleton to the extracellular matrix, which were the CAM proteins, if you remember the CAMs, you can review the previous video, the cell number 4. Those CAMs not only helped attachment of the cell to the surrounding extracellular fluid and matrix, but they also helped attachment to the neighboring cells. The cell membrane proteins help cell-to-cell attachment. So other than the CAMs, we have other structures called desmosomes and gap junctions and tight junctions that help anchoring cells together through protein filaments that pass through proteins in the cell membrane, and we will see this later on. So we have the CAMs. If you're ever asked about, it's true that we have split them into 4 and 5 because 4 is a very characteristic separate entity. But if you're ever asked about attachment of cells to each other, you will say CAMs, desmosomes, gap junctions, and tight junctions. CAMs are very peculiar because not only do they indirectly help attachment of cells to others, but they help attachment of the cell to the extracellular matrix. Number 6, we have cell-to-cell recognition, and we have talked about the glycoproteins and the glycolipids. You can also add here the glycoproteins and the glycolipids that act as flags that help identification of cells by other cells. And now we are going to, we have explained CAMs in video number 4, and now we're going to explain in detail the desmosomes, the gap junctions, and the tight junctions. And this would be an answer in a free response question that asks you about cell communication. But we have different types of cell communication. We have physical communication, the one we are going to explain today through attachment of one cell to another. But we also have physiological communication, meaning that the cells might be very far from each other, but they send molecules to each other to signal or to alert each other so that a certain function or a certain change can be achieved. The first example of physical cell-to-cell attachment is the junctional complex. And you can see the different types here on the diagram. The junctional complex consists actually of different types of connections between the cell. So it consists of the zona occludens or the tight junctions. This is the tight junction, okay? And it also consists of the zonula adherens or the adherens junction, which is the second one. And then the gap junctions or the nexus. And finally, you have the desmosomes. And the desmosomes have another name, which is macula adherens. In a multicellular organism, if we are talking about multicellular complex organisms, the cells must be attached to each other in order to work in a sort of harmony. The junctional complex is an intercellular attachment structure, meaning that it is an attachment structure between the cells. And this junctional complex is usually found around the apical membrane of epithelial and endothelial cells, because you know that epithelial cells are cells that cover the surface of the body. Endothelial cells are cells that line the cavities. Whether they are lining cavities, endothelial cells, or covering surfaces, epithelial cells, they will rest on a certain membrane, which is called the basement membrane. So the basement membrane is the membrane on which they rest, but the other side is called the apical membrane. So these structures of the junctional complex will be present towards the apical membrane. And they form together strong physical connection between adjacent cells and establish communication between the adjacent cells. So this is the first function, is that they establish strong physical attachment or communication between the cells. But meanwhile, it also limits passive diffusion of fluids through the membrane, because if it happens that one cell has more water than the other, water will diffuse or pass by osmosis from one cell to another. In order to regulate this in certain situations, we sometimes need this. We sometimes need the flow of water from one cell to another, like, for example, when water flows passively by osmosis from one root cell of the plant to another root cell, and then through the stem cell upwards and so on, so that the water can reach the leaves. But in some other contexts, we might want to limit this passage of water. So in areas where we want to limit the passive diffusion of fluids through the cell membrane, you will find this complex, for example, in the small intestine. And meanwhile, they limit the passage of fluids or control the passage of fluids, they will also limit the passage or control the passage of materials, solid materials, across the cell membrane. And as we said, they're always towards the apical membrane, and therefore they're always towards the cavity or the lumen if we are talking about endothelial cells that line an organ like the endothelial cells that line the small intestine. And because they are apical, they will be towards the cavity or the lumen of the small intestine. So we call them juxtaluminal in position. And they succeed in order from apical to basal. So this is the most apical and this is the most basal.